fibers are different and can be actually more complex than we know. They're defined in many different ways, but in the microbiome field, we often talk about them in terms of fermentable versus non-fermentable. And that refers to if you eat it, your body can't do anything with it, but your bacteria can do the fermentation that you might get in your kimchi or sauerkraut, and then break it down and then create energy, create metabolites that are beneficial for you. Non-fermentable fibers you tend to eat and they go through you, and those are the ones that create bulk and promote that motility, which is also equally important. As I talked about earlier, you want to have flow daily through your intestines, and so you need the bulking agents and you need the microbial accessible fibers together, and likely blends are the most important. One single fiber probably isn't going to do the trick for optimal metabolite production and just overall gut health.
I'd like to take a moment to thank one of the sponsors of the show, and that is One Farm. They make a lot of great products. It is a farm to supplement company that is really designed to improve people's lives with whole organic ingredients that are sourced directly from the farmers that grow them. You will know where your product is grown, how it was grown, when it was grown, and most importantly, who grew it. Right now, they have a new product, and actually it is a gut health product. I am highlighting gut health in this episode, and they have a bone broth that's been enhanced with botanicals and adaptogens to help support a healthy microbiome and support better gut health. And you've learned all about why gut health is important, which is one of the reasons why I've chosen this product. It tastes great. It's really well formulated from grass-fed organic beef. It has garlic and onions that is grown organic and heirloom. This month, One Farm is offering my listeners a free gut health superfood. All you have to do is pay $5 for shipping. That is a free tub. Go to onefarm.com, add the gut health superfood to your cart, use Lyon GH, that's LYON, and GH is for gut health, to redeem. You can also click the link in the show notes. While you are there, you can check out some of their other amazing products. Suzanne Devkota, thank you so much for being here. I am thrilled to have you, and people are going to get so much out of this. You are a gut microbiome researcher. But we've known each other for how long now?
A while. We have a lot of overlapping, history, and a while.
Isn't that amazing?
Yeah, it really is.
So you did your undergraduate at the University of Illinois, and you did a master's in Dr. Donald Lehman's lab at the University of Illinois.
Yep.
Then did your Ph.D. at the University of Chicago, which for people listening is one of the top programs. And then you did a postdoc at Harvard. And now you are the head at Cedars-Sinai of the Human Microbiome Research Institute.
Correct. Yep.
Wow. Gut health, microbiome, that is the hot topic.
For better or worse, yeah.
And how long have you been involved in gut microbiome research?
Since about 2007. So, I don't know, 15 years almost now, which is scary to actually put a number on it.
Wow.
Yeah.
And, one of the reasons why I'm so excited to have you on and talk to you about it and the gut microbiome in general is you have a very unique perspective. And in the medical community, and I think in the research community, there's the gut microbiome people, and then there's the nutrition people.
Right.
But very rarely is there an individual that interfaces the two. And I'm sure that you've seen that.
Yeah.
That perspective of actual clinical knowledge and academic knowledge of nutrition and the interface of the gut microbiome makes you a unicorn.
Thank you. Thank you. I wish there were more nutrition scientists in microbiome research. And because diet is so important to shaping the gut microbiome, and in the microbiome field, we embrace that, we appreciate it, and we're trying to do nutrition research sort of learning on the fly. And so nutrition folks, get into like hardcore microbiome sciences. Like we need good nutrition people in that space.
You heard it here.
Yeah.
If an individual is really interested in nutrition, do you think that the movement into gut microbiome is really the frontier?
Yeah, I do. I think there's still so much in nutrition to explore, right? That is independent of the gut microbiome. So that is, I think diet and exercise research is having a renaissance right now. And the microbiome is one piece of that I think could fit nicely into nutrition research. But also for nutrition researchers who maybe want to break out and do something different, I think going into the microbiome space would be a great, there's so many niches, opportunities to fill in there where the contribution could be really unique. So both ways, I think it could go.
The obvious question is, what is the gut microbiome?
So the gut microbiome, technically speaking, is all of the microorganisms in your GI tract. So it's not just bacteria. It's your fungi, your protozoa, parasites, all the organisms that are not you, and their gene content. So microbiome refers to the gene content of all of those organisms. If we want to just refer to the names of the organisms, like this guy's here, that we usually say microbiota. And so this is this nomenclature nuance within the field, but microbiome is really encompassing everything in your GI tract, not just the bacteria.
Wow. And that gene content plays a role in human health?
Yeah. We have, in humans, we have 22,000 unique genes in the human genome, which is not that much. The microbiome has 3 times that. 3 to, I think even more than that. I don't have my slides in front of me. But the genes and microbes are all functional genes, and they're doing stuff and producing stuff within our gut.
So you know what that is?
We only know a small fraction. We just know, for as much as you hear about microbiome research, what we actually know is still so small. And we can go into why that is. But we're just scratching the surface. So from what we do know, we know that those bioactive components do affect numerous aspects of human health. And then if you consider the whole unknown fraction, my God, what else could there be, for health and disease? There's so much yet to understand.
So the gut microbiome is the bacteria, protozoas, fungi, and all the bugs, all the other living things and their gene content. Yeah. Whereas the microbiota is just the name.
You're just identifying the organism.
So what does it do for us? And that, when we think about the microbiome, is that just related to the gut?
No. So, you have a skin microbiome, you have a nasal microbiome, an oral microbiome, you have a urogenital tract microbiome, so mostly vaginal is mostly studied, and then gut. So what do all those things have in common, right? They tend to be moist mucosal surfaces. So your microbes love warmth and they love moisture. So you tend to have, in terms of the skin microbiome, more microbes, in the crook of your elbow, behind your knee, behind your ear, than you will just on your dry parts of your skin. So, those are where microbes like to be. But those niches that I just talked about have been evolved since humans evolved on Earth. We became, air-breathing humans. So those are defined niches. But, people have, including our own lab, find bugs in areas that are not those, that are outside the normal niches. And that's when things start to get really interesting.
Okay. What do you guys do in your lab?
We,
Wait, what don't you do? And by the way, I think my daughter has her own unique biome behind her ears. So I'm going to have to
we can swab it.
Talk to the nanny about that one.
The lab has evolved over time. We historically, and we still are interested in diet gut microbiome interactions. So how do the foods we eat lead to pathogenic processes, usually related to chronic disease, either inflammatory or metabolic, through the gut microbiome? And then the flip side is, okay, if we know how diet can cause disease or influence disease through the microbiome, how can we then, reverse that or treat it? Or how can we use diet beneficially for health? So I always say, by studying disease populations, you're ignoring the huge, vast population of healthy people that will also benefit from that understanding. So we try to study both. So diet microbiome is a big focus. And then because of where we are in a hospital and we have access to patients and human specimens, and it's just very easy to ask really interesting questions in human disease, we have a longstanding interest in chronic intestinal inflammation like Crohn's disease and ulcerative colitis. And so we get a lot of these patient samples. Really grateful for patients who want to give up parts of their intestines when they have surgery. And we're actually discovering and still learning that bacteria leave the gut pretty readily in healthy people and in diseased individuals, and they go to other sites in the body and can wreak havoc, essentially. And we're interested in the adipose tissue, so fat tissue, and how fat and the gut interact, kind of like a gut-fat axis. And we're finding that bacteria leave the gut and embed themselves in the fat, and it's associated with a very interesting phenomenon that causes the fat to grow in chronic inflammation. We don't yet know.
As in, when you say grow, you mean would an individual, if I'm understanding correctly, would they actually gain weight, gain fat tissue, or the fat in and of itself is expanding, but the weight is still the same?
So we don't know. I mean, theoretically, based on what we see as to how the fat tissue is morphing, essentially, and growing in this particular context of Crohn's disease, we also have a new project where we are asking that question in obesity. So in obese individuals who are undergoing bariatric surgery, gastric bypass, we get parts of their intestines and their fat, and we're actually seeing if the same bacteria are in the fat of obese people in the same way that we see in Crohn's disease. Or is it disease specific, really unique to Crohn's patients? If it's true that this is also happening in obesity, and we have sort of a mechanism for how bugs and fat and immune cells might be interacting to cause expansion, there could be another layer to obesity.
Wow.
And we're trying to understand that better.
And that's really interesting. Beyond calories in, calories out, which obviously we all can agree, you and I can agree that plays a huge role. It sounds like there's other inputs that can actually not just affect that balance, but actually affect the adipose tissue.
Yeah. And one of the distinctions we're making is visceral versus subcutaneous adipose tissue.
And visceral fat, for the listener, is fat in the abdominal region around organs, and then subcutaneous fat is fat under the skin everywhere else.
Correct.
And what about fat in the skeletal muscle? Has anyone ever looked at that?
I mean,
steatosis?
It is such metabolically an interesting question. As far as I know, has not been even touched in terms of the microbiome. So what might the role there be? We don't currently see bugs in skeletal muscle.
Probably great.
But their metabolites can circulate, so maybe there's some, indirect effect there, maybe. It's an interesting question.
Yeah. It's the way of the future.
Yeah. Exactly.
So in terms of the gut microbiome, you had mentioned you're really interested in the nutritional aspects of the gut microbiome. What could a individual do at home to, there's this term, leaky gut health. Well, before I think we even jump into that, can you define those for me and what your perspective that is?
Yeah, I can try. so much of the microbiome and how your gut your own gut interacts with your microbiome is so personal. I mean, there's so many lifelong influences that determine, so gut health, right? That is there's no, I mean, there are some universally, we know that, okay, the absence of inflammation is healthy, right? But what is the normal homeostatic state? One of the things that we talk about are, okay, let's talk about bowel movements, right? Ideally, you have a bowel movement every single day, right? You want to keep things flowing through the gut. That's a marker of a healthy gut is, you don't have what's called stasis. Things just, because the more your bugs kind of stay in one place, they will multiply, and then that interacts with your gut cells and can cause localized inflammation. So you want to keep things moving for multiple reasons. Also, so you feel good, you don't feel bloated, all that sort of stuff. Things like, stool consistency, we can talk about that. I mean, everyone, frankly, should look at their stool, and it is an early indicator of many diseases if you have something off there. And you should talk to your doctor if you have really open conversations about bowel habits and all the observations you make, because there's a growing body of evidence suggesting that so many early indications of even neurological diseases can manifest in the gut. And so,
and would that be like constipation?
It can be some kind, or either or. Constipation can often be a sign of some nerve damage, right? Because you're not, the motility aspect is not working properly. And so there's a strong link between the gut and multiple, organs, systems in the body, and therefore the diseases that affect those systems. So the gut really is so important to really pay attention to, and you only know your body, and so when something's a little off. So gut health is really personal and it's unique to you, but those are some things that sort of are universal things you ideally want to strive for. Leaky gut is kind of a problematic term, but it's a real thing. So, but how it's being used is probably a little more loose than what it actually, like how you define it, right? So in the lab, how we define leaky gut, or we call it intestinal permeability, is the more, what I feel comfortable describing it as. Your gut is lined with cells, and those cells, when you have chronic inflammation, or if you shift your diet to one that might be more immunogenic. And the Western diet, which is a big umbrella, has been pointed to as an immunogenic diet that causes gut barrier problems. With that, the junctions between the cells that hold them together become loose, leaky, okay? And bacteria themselves, bacterial products, food antigen products can pass through those cells into your bloodstream. You don't want that to happen. Typically, you don't want like, your gut is the outside of your body. So your mouth to your anus, that's outside. So it can handle a whole bunch of foreign antigen pretty well, but that barrier is there because the rest of your body can't. And so you don't want that seeping out. And so that's leaky gut, and we measure it through actually taking intestinal epithelial cells, the cells that line the intestine, and measuring those genes. And if they are downregulated, that's usually a sign that there's a problem. Yeah.
And could someone at home or with their doctor look at markers like zonulin for if an individual had leaky gut, which you so beautifully explained? Mm-hmm. So essentially it's the intestinal lining, those tight junctions are coming apart, and then things are getting in there and then getting back into the bloodstream.
Yeah.
Is that
so? And it's there are many papers trying to correlate zonulin or things like secretory IgA that are often measured in gut health tests to the actual state of leakiness through the means that I've just described. And some will show really tight correlation. So I think there could be something there, and then others show inconclusive.
Interesting.
And it really just depends on the context. So I don't think we should discount it. I think there could be utility to it, but I just can't really confidently point to it being as a definitive biomarker.
So how does somebody know if they have leaky gut?
It's a good question.
How do we know if we have intestinal permeability?
Yeah. Leaky gut tends to just not occur like spontaneously.
There's no spontaneous leaky gut occurrence.
As far as I know, that it's just something you have to do something for that to happen. So if you know you eat a poor diet, okay, again,
and that would be processed foods,
a lot of food additives, probably a high sugar diets.
Do food additives affect the gut microbiome?
Yeah. Certain, I mean, this is a really hot field right now, and certain ones have been pointed to, and there's some really interesting studies. Right now, it's like there's a study looking at one, there's papers on a single food additive, and you string them all together, and maybe
Can you share which. Is there one in particular?
Some of the ones that have been recently studied were, I think, polysorbate 80 is often in products. I think that, leaky gut permeability is one readout often for these things, and they tend to lead to things that affect gut permeability. The other thing they can often cause is a bloom or an overgrowth of certain bacteria that can cause problems.
The food additives can feed. Would you call them bad bacteria, or they are just?
We have various names for them, pathobionts. But an example, there was a study that came out looking at the food additive. I can't remember off the top of my head, but I just remember that it's in Taco Bell meat, and it actually
nobody should be eating Taco Bell meat. I'm just kidding, because it's probably not meat. We don't even know. It's probably crickets.
It causes like a slight sweet flavor in foods, and it's in a lot of foods. They'll come to me, I'm sure, as we're talking. But they found that it can cause a bloom of Clostridium difficile, which, and people who are susceptible, you probably don't want.
Nobody wants Clostridium. No one wants C. diff. For anyone who is listening, C. diff is, if you've worked in a hospital, you know there is the C. diff smell. It is dangerous. It is robust diarrhea.
Right. Yes. It's pretty bad. So there have been studies showing that where, and there, some of these are a bit reductionist studies, and some of them are only in mice, but the data is compelling, and there's enough sort of pieces of evidence that have kind of similar readouts showing that these things are making a difference. But you always got to look at the statistics and look at, is the delta really big, or how much do you have to eat as a human to actually have that effect? Taking a mouse, what affects a mouse and applying it to a human could be very different things. So we had to we should take them as, suggestive and paying attention to them, and more work needs to be done. But there's probably something there.
And I think one of the things that is really compelling is we talk a lot about, especially in the nutrition space, we talk a lot about macronutrients. And then we talk about the processing of food, whether a food is highly processed, ultra processed. Very rarely do we then move to the food matrix and the forced food matrix of foods that don't exist in nature.
Yeah, right.
And there is a huge gap between what you're talking about as a gut microbiome researcher and our perception of health and wellness, that we don't even know what the food additives are doing. I'd like to take a moment to thank one of the sponsors of the show, LMNT, LMNT. When we are talking about gut health, we're talking about nutrition and hydration. LMNT is a tasty electrolyte drink mix. And in fact, as we go into winter months, I may or may not have it in a hot glass of water. It has 1,000 milligrams of sodium, 200 milligrams of potassium, and 60 milligrams of magnesium. If you are trying to keep your weight low, this is an amazing strategy to manage hunger. I think that this is amazing, especially if you are training hard and sauning a lot. There's no junk, no sugar, no coloring, and nothing artificial. It doesn't have any fillers, and it was formulated to help everybody. It doesn't matter if you are keto, low carb, high carb, or paleo. Right now, LMNT is offering my listeners a free sample pack with any purchase. 8 single-serving packets. Great way to try all 8 flavors. Get yours at drinklmnt.com. LMNT, DRINKLMNT.com/Dr. Lyon. LMNT offers no questions asked refund. Try it totally risk-free. If you don't like it, they'll give you your money back. One of the other things, and I know that people are really wanting to know how they would be able to identify their own health, their own leaky gut. And one of the things I get actually asked a lot is artificial sweeteners, which I think would be in a line with food additives, but
Yeah, it is.
Technically speaking, we have aspartame. Do they even use saccharin anymore?
I think rarely.
Saccharin, Splenda, dimethylene aspartame, probably monk fruit, but that's not artificial sweetener. So how do artificial sweeteners, do we know how they impact the gut microbiome? Just throwing you to the wolves.
I know. Yeah. So there is a growing body of research looking, it's still under that banner of a food substitute similar to food additive in the sense that these are non-natural things that have been created and put into food products. So very, again, hot area of research. My first reaction is I don't believe they have. The data doesn't convince me that they have a overwhelming effect on the gut microbiome in a detrimental way. But what I always say is no matter what we're talking about is anything in excess will have an impact on your gut microbiome. So sugar, for example, gets absorbed pretty high up in the GI tract, usually doesn't have an effect on your colonic bacteria. If you eat enough of it, it exceeds the capacity for your body to digest it. It will reach the colon, your bugs will see it. So there's always a question of quantity. And I think I always, encourage people to exercise a little bit of caution around black and white statements, and that, one pack of Splenda in your coffee each day is very unlikely, even without seeing the data, I can tell you, very unlikely to have an impact. Putting, a tablespoon of heavy whipping cream in your coffee even, not going to have that much of an impact. But if you're putting, if you're drinking 10 cans of Diet Coke a day, there's likely going to be maybe some effect because that is a lot of anything in your system. And that goes for any food. And so it's everything in balance, right? And you shouldn't deprive yourself, but you shouldn't do anything in excess. Now, one of the things I worry about in the messaging around artificial sweeteners in the gut microbiome is what's the alternative, right? So does that mean you should be going back to drinking normal Coke, right? Full sugar things again. What exactly is the byproduct of that message? And I don't think that's the answer either. So we cut it out completely. Maybe that's not easy for everybody. So we have to, think about, as we sometimes demonize certain things, what the alternatives are, and is that alternative better than the other?
Is there a food or food product you would never touch? She's not going to say bagel, folks.
Yeah. Wow. I will eat, I have eaten just about everything in various parts of the world. So in terms of that, but what do I choose to avoid? I'm generally low carb eater, but that's not because of the microbiome. That's just because of, other metabolic things. But I do, after a lot of the research has come out, I really do try to avoid as much processed food. It's impossible in this world, everything is processed to a degree unless you're having a hunk of meat and a vegetable that you grew yourself. But I try to limit the processed foods because I do think that chemicals can get to your gut microbiome. Your body can't absorb them in the same way, and so there are those effects I think that are possible. And I try to avoid sugar as much as I can as well. But I don't, I personally consume artificial sweeteners, and I have no qualms about it. But I do it in moderation, not that much.
What about colors? as a food additive?
It's interesting. I think there's been one paper on the red color, and found some interesting, but red, like the color you'd think that they're inert, but one paper showing that there was an effect on the gut microbiome and, of course, showing detrimental effects. And I can't remember if they were looking at a metabolic or inflammatory disease, but whatever it wasn't good. And so you could extrapolate that to all the colors, and say, we just don't know. When all these food additives and preservatives were introduced, it was long before we were really studying the microbiome. Right. They've been in our food supply for so long.
Would you think that was from where, during World War II times? And Industrial Revolution?
You have Spam. You have there's a reason for nitrates in meat. I would avoid those, but not avoid meat, but avoid the preservatives and almost anything.
And also the naturally occurring nitrates as well. Do you think that?
I don't.
Is there a difference? I guess is a better question. Is there a difference for some of the things that are naturally occurring as a byproduct from curing and things of that nature versus?
I don't know. I honestly don't know the answer to that. It might be a quantity thing. But I don't know. Chemical structures are chemical structures, right? So theoretically, they should be the same. And we like to think things from nature are better than things that are added later, but a chemical is a chemical. So I think it's a quantity question.
When you think about things that would totally wreck the gut microbiome, and again, we are talking about the bacteria, all the things, and the genes, which is, I'm curious as to how that even incorporates into our health and wellness. If you were to think about one, the top 3 things that just would wreck your gut microbiome, that sounds like it's processed foods.
Yeah.
What about alcohol?
So, oh my gosh, okay. I think I tread carefully here. So alcohol is probably one of the most overlooked pieces of data in microbiome research. So when you do a study in humans, you take a survey, like of people's lives, and what do you smoke? Do you drink? Do you have family history? All that sort of stuff tends to go in like the basic information when you're recruiting people. But that information often doesn't get rolled into how we interpret the data. And so there was a paper that came out 2 years ago from actually a colleague of mine who is now at Cedars. And he published it in Nature, and it was a computational study where he took a bunch of data sets and looked at the most determining lifestyle factors, or human factors that affect your gut microbiome composition, and then what was actually reported in the papers. And when he did all this analysis, he found the biggest drivers of gut microbiome changes in composition were alcohol consumption and bowel frequency and consistency, which I talked about earlier. And those are things that are often not reported even if you're not studying alcohol, it's not reported in the data as a potential influencer or confounder. So it's sort of, people have been talking about that and saying, okay, well, A, we probably need more alcohol studies to really understand that. And 2, we need to start reporting and recording this data because it could affect what we're seeing. So the short of it is we don't really know, but it seems to have an influence. And I can't tell you if it's one type of alcohol versus another, which is usually the next exciting question.
Yep.
And, fermented versus, degrees of fermentation. And, wine and beers we don't typically consider those fermented foods in the same way we do kimchi or sauerkraut or kombucha. But there are metabolites in those as well that could potentially be beneficial, polyphenols and things like that. So we shouldn't ignore those. So maybe I would say that over mixed drinks, but that's for metabolic, sort of reasons. But there's just a lot we don't know yet.
And, by the way, we are drinking kombucha, aren't we?
We are drinking kombucha.
Seasonal kombucha, yes. Which I do want to talk about seasonal foods and how they affect the microbiome. Before that, though, what about fermented foods?
Yeah.
And you and I had a very interesting conversation in California, and I was asking you all about fermented foods. And and the probiotics on the label. And you say, Gabrielle, it's not about the probiotics on the label. It's actually the metabolites of the fermentation.
Yeah. Yeah. This is still, these are kind of emerging concepts. So a bottle of kombucha is a great example, or many of these fermented foods will list the bugs, right, on the label that are in that particular product. And they're marketed often as probiotic drinks. And I think the vast majority of people kind of associate probiotics with good, like beneficial, or at worst, I mean, we don't know, but at worst they do nothing, but potentially beneficial. And what isn't often distinguished is sometimes those probiotics are just added after the fact. They're not actually the bugs doing fermentation in the bottle, because if you're pasteurized, you kill all the bugs and you add bugs back.
Oh,
so it's kind of, in my view, a little bit kind of like cheating. That being said, doesn't matter, because that fermented product is a product of a living process. So fermentation creates some known primary chemicals that are really good for the body, but there's a whole slew of thousands of other chemicals that they produce at different quantities that we know and have studied, we as scientists know and have studied, as affecting multiple processes, like scavenging free radicals, antioxidants. There's things like serotonin-like chemicals in them. When you drink it, I can't tell you how much of that gets out into the bloodstream.
That was my next question.
Yeah,
Okay.
I don't know. But many of those can act at the level of gut for health and for improving things like we were talking about leaky gut. Those can help keep the barrier, the junction closer together.
Because of the chemicals, or because of something that it's doing within the, the environment of the other microbes?
It's direct effects with it could be also influencing the microbial communities, but actual direct effects with your intestinal lining.
So the fermented foods don't actually, or do they actually affect the gut microbiota? It sounds like that's not the primary driver. It sounds like the chemicals or the compounds that are made in the fermentation process are doing things like acting as an antioxidant, also having some kind of, or component, this might be a huge jump, of some kind of healing capacity towards that intestinal lining.
Yeah.
Am I hearing that right?
It's more of that direct effect of the chemical and your body, right? Not so much through influencing the microbiome composition.
I think that's fascinating, and probably the opposite of what people are talking about.
Yeah, it's very different. But so that's what is in the bottle, right? Now imagine if you could encourage all of that to be happening naturally all the time throughout the day in your gut, because your bugs have that capacity to be like a bottle of kombucha, right? To be actively producing all those metabolites, so you have a renewable source of it all the time, and encouraging those chemicals to be produced and to improve your gut health or maintain your gut health. And that's pretty, like, we talk about things like short chain fatty acids produced by gut bacteria. We know those improve that barrier, your intestinal barrier, but you have to feed your bugs in order to have that produced.
And what kind of foods would you
primarily
feed that?
Fiber sources are the primary driver of like short chain fatty acid production, but other chemicals.
Any kind of fiber?
There's fibers are different and can be actually more complex than we know. They're defined in many different ways, but in the microbiome field, we often talk about them in terms of fermentable versus non-fermentable. And that refers to, if you eat it, your body can't do anything with it, but your bacteria can do the fermentation that you might get in your kimchi or sauerkraut, and then break it down and then create energy, create metabolites that are beneficial for you. Non-fermentable fibers you tend to eat and they go through you, and those are the ones that create bulk and promote that motility, which is also equally important. As I talked about earlier, you want to have flow daily through your intestines. And so you need the bulking agents and you need the microbial accessible fibers together. And likely blends are the most important. One single fiber probably isn't going to do the trick for optimal metabolite production and just overall gut health.
And translating that to the human diet, that would be varying your food content.
Yeah.
Right? It wouldn't be just eating apples to get pectin over and over again.
Correct.
You want to have whatever you eat, apples or grapefruit. It's really varying that content. Yeah. The fermentable items, is that, when you say that, what would an example of that be? Is it just kimchi and those kinds of actually fermented type products, or is there other foods?
So in terms of the fermented foods, we always point to, kimchi, sauerkraut, kombucha, fermented dairies like kefir,
which we eat all the time.
Yeah.
Yep.
All those are great as well. Pickles, some are fermented, some are not. So there's a little bit of, I think, misunderstanding that all pickles are fermented. That's not the case. You want to look for ones. Pickling can just sometimes just be salt and vinegar, and other times they actually have bacteria that help ferment, and they're actually really good. It creates a good flavor, but you want to look for that on label. So pickling doesn't always mean fermented. Just want to make that disclaimer. So that's the fermented foods, and there's others out there, and people are getting really creative with fermentation. But fermentable fibers are fibers that you can eat. So like things like your Metamucil will be a bulking agent, right? And you take to help move things along. Microbial accessible fibers will be the one we see often on labels is inulin.
Inulin, yep.
It's a great fermentable fiber. It also causes a lot of bloating in a subset of people, so not everyone can handle it. But there are other types of fermented fibers. We're talking about pectin. We're talking about the green banana, the resistant starches. All of that can be really beneficial as well.
And should people, if they were to think about proportions, should they include a resistant starch every day? I guess the bigger question is, how quickly can we influence the gut microbiome in any kind of lasting way?
Right. With
the fermentable foods, with the inulin, with the cellulose. And you've done some very interesting work with some of these fibers, which we will definitely talk about in terms of being able to affect nutrient deficiencies and leveraging the gut microbiome to address nutrient deficiencies.
Right. Yeah.
So how long does it take to influence the gut microbiome in a meaningful way? Through diet first.
Right. Yeah. So we have done some studies that we've looked at intermittent fasting and done, putting food in, taking food out, and how you can affect gut microbiome composition over time. We've done this in mice and humans. You can, if you withdraw food, let's just take the extreme case first, a fast, right? Within 18 to 24 hours, you can radically shift your microbiome. But as soon as you introduce your same diet back, it rebounds. So on 24-hour scales, you won't change. I mean, you can change, but it'll revert. So when you're talking about long-lasting change, you need consistent food patterns. I mean, how long exactly? I think no one really knows the answer to that. When we do diet studies, for us, the magic number is about 3 weeks, whether it's a mouse. So a mouse, it's about 3 weeks. A human, we always say 6 weeks for,
I was just thinking double that. Yep.
Yeah. Which kind of makes sense. But I have colleagues who they've been case studies for me where they internationally travel a lot, pre-COVID especially, and they're really fascinated by their microbiome. So they would give me a stool sample before they went to...
But let's just clarify, they wouldn't directly give you the stool sample.
They would leave it in like a baggie.
Suzanne loses all of her friends every time they go on an international flight. But
Hey, it is fascinating. So we get these magical samples that just appear. I don't look at them.
She doesn't know the name.
I don't know who. But before and after a trip, right? And so we have all these data points from different parts of the world, and I can after a 2-week trip to Korea, I can see meaningful changes. But it'll revert back.
Meaningful meaning you see an influx of good bacteria. How do you define meaningful? How do we think about it as it influences the health of the individual?
So, the first thing we're looking at is just a shift from before. What is the delta? What is the difference from where they were to after the trip?
And is it beneficial to go through periods of time where you shift your microbiome?
That's a really good question. I don't know. My instincts would be no, unless you have issues, right, that you're trying to correct or fix, or but if you have an otherwise healthy microbiome, your bowel function is normal, then I would say no. You don't want to create perturbations in your gut microbiome. It's not like doing a cleanse, right? Colonics and all that. I don't think they're necessary. But
could they be helpful for those? Do you want to explain what a colonic is?
You should explain what a colonic is.
Darn it. Look it up.
Yeah. I think there are times when, yes, you might need one, but I think proactive shift in your microbiome is unnecessary if it is otherwise healthy. But there are times when I do think things go awry. So definitely during a lot of international travel, I think most people can attest to the fact that not only is there sleep off, which can affect the gut, by the way, all your bowel movements are wonky. When you travel and when you come back, it takes a while to get back into a rhythm. That is affecting your gut microbiome and vice versa.
Can I stop you?
Yeah.
Is that okay? So I guess the obvious question would be, is it related to the change in food, or do they have their own circadian rhythm? Mm-hmm. That might be a really not obvious question, but do they have their own circadian? And when I say do they, we're talking about the non-us living organisms that are going along for the ride.
Sure.
Do they have their own circadian rhythm that changes, and does it matter? I guess the question I'm really trying to ask is, how is that environment affecting the gut microbiome, not even related to the food that we're eating? Because, what if, let's say, so I used to pack all my food with me, right? And I would travel. And even if I packed all my own food and didn't ingest anything outside of what I brought and what I normally ate, I would still not feel great.
Yeah. 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 Those are great. Those are great observations. And so the circadian microbiome, axis and the effects systemically is a really exciting and hot area of research right now. And some things that have come out of studies that are compelling are over a 24-hour period. So we have a light-dark cycle as living as mammals.
Well, I don't, because I have a infant and just kidding.
I get it.
Yeah,
I get it. You'd actually be a great case study yourself. So your microbiome naturally goes through some populations will go up and down over a period of 24 hours. And it's a cycle that appears to be somewhat predictable. We don't know why that is, but whether it's influenced by your body, putting some pressure on your microbiome to go up and down, or if the bugs just naturally have their own, cycle within.
Do we know how long they live for?
The bacteria?
Yeah.
They're turning over. They're turning over constantly.
Got it.
They're not very long lived at all.
So there's not Norma in your gut that's been
been there for 80 years. Yeah. No, huge amounts of turnover. But they clonally expand, so they have clones, right? Mm-hmm. So the original founder bug may have been long gone, but exact replicates exist of them. So, and then there's been studies showing that, if you give a mouse antibiotics over, the course of a week, they can lose their circadian rhythm in certain organs. So we think of our sleep-wake cycle, your liver, has its own circadian rhythm that is separate from your brain. And apparently, the gut microbes can regulate some of what's called your peripheral clock. And that relates to diabetes and obesity and is studied in terms of metabolic diseases. So does the gut microbiome have their own circadian rhythm? It seems perhaps. We don't know what in the world that means, or what's chicken or the egg. But to your point, your sense of, when you travel a lot, food can help kind of reset, but it can't do all the work. And so your biology and being off cycle I think has an important influence.
Can the people that you live with influence your gut microbiome?
Yeah, yes, there's some really interesting data there too. So the environmental microbiome is a whole area of research, and that's called the built microbiome. And that typically just refers to hard surfaces, right? So there's a hospital microbiome. So they take swabs of new hospitals and old hospitals and see how microbes live in the air. They live on the walls and they live around us in abundance. And that goes for our bodies, right? So we have bugs, yes, in our GI tract, but we walk around with what's called like a microbial cloud around us, kind of like in Snoopy or Charlie Brown.
What is it? Is it Pigpen?
Pigpen, yeah. Pigpen. That's the image I often will use, is that. So we all have kind of that. And it's a mix of just dust, clothes particles, skin particles. When we breathe, it's just our unique aura. And people who cohabitate, and not just cohabitate, but so there is a study, which I like to refer to sometimes, where they surveyed married couples. And they looked at people who cohabitate who are not married, people who cohabitate and are married and describe themselves as having a very close relationship related to intimacy, and married couples who maybe are not as intimate. Those who were married and closely intimate had very similar microbiomes to those who just cohabitated and maybe were not having as much physical contact. So simply cohabitating it's almost like a gradient, right? So you will share, dishes, things, you're in each other's space. It also depends on how big the space is. But if you are sleeping in the same bed, sharing a lot of the same things, you will have a more similar microbiome than your neighbor next door, and so on. To the point where, and this will be another topic I'm sure we'll get into, with fecal transplants.
And you have to explain what a fecal transplant is, because it's, I first heard about it a couple years ago.
Yeah.
And I would love for you just to take a minute and explain the concept of a fecal transplant.
Sure. Well, it's exactly like what it sounds like.
Yes, it is.
You are taking healthy stool from a healthy individual, and you are being reseeded with their microbiome.
And is that done? The fecal transplant, it's encapsulated, isn't it?
There's many protocols depending on where you do it. There are capsules that do it, so, and it's like a 30 capsule dose, or there's a 2-route introduction, so a nasogastric tube, and through the anal route, so you get the top and the bottom.
But it's washed, right? It's processed. The fecal transplant is processed in some
Yeah. I mean, it's tested, first
Okay.
of all, to make sure there's no communicable diseases, pathogens,
Right.
first of all. And then most of the time, and I'll get to that in a second, which is why I'm get back to the cohabitation part. So there's that. And then, yes, it is prepped lightly. So it's kind of spun down and kind of filtered to a degree, but it's really interesting because people have tried to create synthetic stool so you don't have to actually use the real thing, and they find it doesn't work as well as the real stuff. So you don't want to.
Why would anyone ever do? Why would an individual do a fecal transplant? And
well,
how did we
not
even
a
get
hobby. It's not a hobby.
And how did we even get here? No.
I know. Well, it's been described in ancient Chinese medicine for centuries, and you can go, and there's been references.
And is it for youthfulness? Is it for the concept of taking a young, healthy, I mean, probably at the time they didn't even think about gut,
Right.
but young, healthy, vibrant essence, I don't know how to say it,
Right.
and then transplanting that into an older individual?
I don't think in the historical text that was the reason why. I think it was actually to treat ailments likely of the GI tract. Interesting idea.
Not evidence-based. I'm sorry.
But today in America, the reason it is used and quite actively in medical practice is for recurrent C. difficile infections.
In fact, isn't that the only FDA approval for it? Yeah.
And unfortunately, because I actually think there's a lot of benefit. So if you go there in Australia, they have far less regulation. And, I hope that continues because a lot of people I know have gone down there and been beneficially treated for non-C. diff related.
For an example, what would the person go down there?
IBS-like symptoms, irritable bowel syndrome,
Which
right?
would be a mix between bloating, constipation, diarrhea, anything irritable.
Yeah, functional issues. So it can be people who, in this particular case that I'm referring to, someone who actually was diagnosed with Crohn's disease as a child, but managed through medications, but then developed later in life SIBO, small intestinal bowel overgrowth, and all the IBS symptoms that could not be managed
with medication. So they tried Xifaxan and
and it was just huge for someone who travels a lot, and was a huge problem. And we sequenced his microbiome and found that because he'd been on sort of a series of antibiotics, steroids, decimated his microbiome. And I made a sort of casual comment to him, hey, you'd be if only we could do it in the US, but you'd be a perfect candidate for a fecal transplant. But you don't have C. diff. He didn't have C. diff in his microbiome. And he just kind of latched on to that, and he searched high and low, searched for clinical trials in Europe, searched around, and nothing he was eligible for in the US nor Europe. And then found a clinic in Australia, who I know well, who you go, it's a 3-week process, and you go, and it's a pretty intensive process. And he went and he did it, and it was like a new person.
Wow.
And we sequenced his microbiome thereafter. They had 4 donors, and his microbiome exceeded the diversity of the donors. And
that's incredible.
Yeah.
How long does that last?
So it lasted until he traveled and picked up an E. coli and had to go on antibiotics.
Oh, he probably was thinking, God, this is the worst case scenario now. Okay, so then did he do it again?
He did it again.
Okay.
And he's actually done it 3 times because in between he's had some infection that has come up that has altered.
From his Crohn's or just?
From environmental, picking up a pathogen.
Do some people have a lower robust gut microbiome just at birth?
It's a great question. Probably, yeah.
It's like, why would this guy have to go back because that's probably not available for the majority of people.
Yeah.
And you would think once you repopulate the gut microbiome, that he would just be able to mitigate any kind of other bug.
Anything,
Yeah.
Like many of us
as to why he's unable to maintain that.
It's possible that he was just, those communities were there populating, but they weren't maybe as robust as they could have been. I mean, you take these transplants and you still have to support them, right? And I think that's an aspect that people are looking into, but we need more research into the dietary support post-fecal transplant. How do you encourage those bugs to grow? And it's probably a mix of fibers and diet, diversity of the diet. And that's often not talked about, even for C. diff. So,
and that's probably, again, that division between the nutritional scientists and the gut microbiome experts or the, microbiology individuals.
Yeah. And that's where, we need more people talking,
The interface between the two. Yes,
exactly.
What would, if you were to think about, I think that we should probably take a moment to talk about sequencing. Mm-hmm. That is something that probably people don't think about in terms of how can we identify what is actually in the gut microbiome.
Right. Yeah. And sequence, when we refer to sequencing, it's the tool, the technology we use for identifying these bacteria. That it is really the microbiome field emerged from infectious diseases and kind of microbiology because of the invention of sequencing the DNA of these bacteria. And we discovered that you don't have to grow them in order to see them. You can actually take a stool sample or a soil sample or an ocean sample, a skin swab, whatever, and essentially run it through a machine, and it will tell you who is in that sample, what bugs are in that sample. And you couldn't do that before 2007, like when I got into the field.
That's very new science.
Yeah, but it's been around for the human genome for a long time.
But for the gut microbiome, that guy definitely won the Nobel Prize. He
unfortunately has passed away several years ago. But that was probably the biggest, thing for the field. And so sequencing now is the bread and butter for microbiome research. And usually it's what we use when we want to pilot an idea. And we want to say, okay, we want to give this diet versus that diet or whatever the treatment group is. And we want to see who's changed. We'll sequence, and then we look for changes, and then we decide where to go from there. And then, at-home testing kits that are everywhere.
Let's talk about it. All of the at-home. And the major goal, right, is that people are trying to understand their gut microbiome.
Yeah.
Which, how many bugs is that?
I mean, like 100 trillion bugs in your diet.
And in a gut microbiome test, the best that we are possibly looking at is what? Maybe 100?
Yeah. I mean, it depends on the technology they use and why it's so challenging for people, like your listeners and people out there who are interested in this. There's probably 50 different companies selling microbiome testing kits for at-home, let's say at-home use. And they all use different technologies that all generate different types of data. Some are better than others, but none of them anymore just say, we're just going to sequence your microbiome and tell you what's there. The vast majority sequence your microbiome and then tell you what to do. And they charge you a lot of money for that. The telling you what to do part is so tenuous and based on very little data.
Meaning someone will send in a gut microbiome test and they'll say, okay, well, you should eliminate eggs.
Yeah.
And they're making a broad, generalized recommendation based on, I'm sure that there's some bacteria that we know are beneficial and some bacteria that we know are harmful, like with C. diff, but there's so many different positive bacterias that there's, in any one family, there's a zillion.
Yeah. And what's beneficial or harmful in one person may not be for the next person.
This is very difficult and very challenging.
Yeah, it's huge. And I think, when people do these tests and they get that information back and they follow it, but it's not personalized to them. Yeah. Very rarely, and mainly because if you look into the literature, I couldn't tell you. And I've been doing this for 15 years, and I could not tell you if you're missing a bug or that bug is overgrown, exactly what food to eat. I can give you suggestions, but I could not tell you definitively how to correct that. So the kits are great in many ways for getting the information about your composition of your gut microbiota. And it's good, I think, to have it when you're healthy before,
I think that's actually a great idea. And then the other part that we talked about before we started recording is that there's secretory IgA and calprotectin and zonulin. Even if those, depending on why we're looking at, we can still track it.
Yeah. Yeah, I mean, and so we need markers like that you can sort of pair with maybe gut microbiome changes, exactly, so you can track it. And so you're healthy now, you have your baseline levels, and then maybe one day you have to go on antibiotics because you picked up something, and you want to know if your gut microbiome has recovered, right? You want to know how close does it look to where you started 2 months ago or whenever you tested. That's important data, I think, to have for everybody. So the kits, while there are some issues with them, having some information at a baseline, in my opinion, is better than nothing.
I'd like to take a moment to thank one of the sponsors of the show, and that is InsideTracker. InsideTracker is an amazing service. We're talking all about gut health. And when you think about gut health, you do think about nutrient absorption. There are certain markers that have the potential to highlight how good your absorption is. For example, iron. Are you consuming enough iron? Are you actually absorbing it? One way to do this is to test and not guess. You can go to inside, like inside your body, tracker.com/Dr. Lyon. You'll get 20% off. Very, very valuable to do this. Go to InsideTracker.com/Dr. Lyon for 20% off their entire store. If you care about health and well-being, then I'm assuming that you do because you're listening to this podcast, and that is the mission here, is to provide you with transparent conversations. You have to know what you're working with, and that is why InsideTracker is so valuable. For a limited time only, you'll get 20% off the entire InsideTracker store. Just go to InsideTracker.com/Dr. Lyon. And take care of yourself. You have to, at some point, be your own best friend and make sure that you are doing the things for yourself that you are doing for everybody else, because without you, none of it matters. So take good care of yourself.